Dr. Ruibin Kuang of the Guangdong Academy of Agricultural Sciences, China. Dr. Ruibin is an associate professor at the Institute of Fruit Tree Research, Guangdong Academy of Agricultural Sciences. She obtained her PhD in biological sciences from the University of Hong Kong in 2009. Dr. Kuang is actively involved in various research involving, including passion fruit, banana, and papaya in aspects such as germplasm collection, evaluation, and utilization, genetic improvement breeding, plant disease resistance mechanism, cultivation technology, and fruit functional components, a vast area of expertise, Dr. Kuang. So she continues to pursue her aim of improving the breeding efficiency, growth, and yield of fruits in South China. In the past decade, she has received 4 research awards, and 5 of her technologies and 10 fruit varieties have received patent application in China. We welcome you, Dr. Ruibin, to deliver your presentation titled Current Trends and Challenges in Passion Fruit Research and Development in China.
Good afternoon, everyone. Firstly, I would like to thank TFNet committee to give me this chance for presentation. I'm honored here to talk about the current trends and challenges in passion fruit research and development in China. The contents involved in 5 parts. Firstly, the importance of the passion fruit, and the second is production and development of the passion fruit in China. And also, I would like to talk about the production limitations, especially the color rot disease as a threat in China, and the pathogen isolation, characterization, and also the integrated control in China. And finally, I will talk about the research respect post-batch of this project. There are 18 general Passiflora family, consists about 530 species, which only 50 to 60 species are edible. Passion fruit is cultivated widely in tropical and subtropical areas in the world due to its edible, ornamental, and also medicinal value. Here is the summary of the passion fruit's functional properties and its potential industry application. The passion fruit juice is used in the food and beverage industry. The material fruit juice is sweet, sour in flavor, and highly aromatic. Seed is equipped with fatty acid, phenolic, and flavonoid. And around 20% of the seed is equipped with fatty acid. Extracted from seeds mostly are unsaturated fatty acid. In the pure insoluble dietary fiber composed of pectin, cellulose, and hemicellulose, it has a long history of the use of traditional folk medicine and can be used formulated as the food supplement for healing disease such as cancer, immunodeficiency disorder, and cardiac disorders. The passion fruit was originated from South America. In China, it was early recorded since 1920s and widely planted since the last decade. It became a new economic and efficient fruit planted widely in South China. The reasons are as follows. It contains rich nutrient contents and easy for fresh juice and processing production. It can be easily storage and it has long shelf life, which is fit for the commerce platforms nowadays. And they harvest in the year of they are harvested in the year of planting and have long harvest period. So it is widely planted in South China with high economic value. Here show the production status of passion fruit in China in 2020. The harvest area was about 53,000 hectares, and the yield is about 1.5 million tons. It was mostly planted in South China, and Guangxi is the biggest production area, followed with Fujian and Guangdong provinces. And Guangzhou and Yunnan are the two new planting centers in the west south of China. There are three main production systems in China. The first is mixed with other crops in eastern fields. They produced on small holdings and with resource poor with the limited inputs. The second is production less to the homestead in the rural areas or in the backyard in the urban centers. Became popular to shape and beautify living surroundings. And the third is monoculture production, which is the widely used production system. It is in intensive production in monoculture. In organic fertilizer are applied. The plots are kept weed-free and disease and pests are well controlled. They produce destined for urban and domestic markets. Here show the four major cultivars in South China. Tailong #1 and Mantianxin are the purple red and fit for the juice processing. Bale, Huangjing, and Dahuangjing are yellow and sweet, sour, fresh edible cultivars, which have higher value than the purple ones. And these four cultivars have rich nutrients and aroma contents. We found the nutritional composition content in purple passion fruit was basically the same as the yellow ones. The two yellow fruits have higher soluble solids and lower titratable acid, and also higher vitamin C content, which make it more eatable than the purple ones. We also found a high potassium contents in the passion fruit, which is comparable to bananas, a well-known high potassium fruit. We also found that about 105 kinds of aroma contents in the purple passion fruits and only 79 in the yellow ones. The highest contents were esters, and then it's alcohol and the alkyl. The harvest standard was mostly decided by the shipping and the circulation market. Here is the criteria for the purple fruit in the Guangdong local market. The fruit size, pure color, and also the soluble solids concentration are detailed required. And a range of passion fruit products has been developed, including jam, jelly, vinegar, wine, yogurt, compound beverage, condiment sauce, and so on. But the deep processing example with the seeds, roots, and wood for their medicinal components was still lack of research. And according to our field investigation, we found some major limitations in passion fruit production in China. The healthy virus-free plant seedling were necessary, and the deep processing techniques and efficient cultivation technology are necessary. And major diseases and pests such as color rot disease and wood beast virus were the main threats for the passion fruit production. Extreme climate effects, such as the high temperature in summer, flower fruiting term, and extreme cold in winter, affect the yield and also the quality. And the above problems have reduced the economic production period and making growth of these species uneconomical in certain regions. Among these, the cholera disease was reported in China from 1990s. This disease starts with polar root growing from a spot to a whole stem rotting, and the branch yellowing and wilt until the whole plant dries. In South China, it is observed in adult plants in spring rainy season. However, in soil with the disease history and higher temperature and moisture, young plants can also die under the pathogen attack. So for the pathogen isolation and characterization, the symptomatic disease plants were collected from different passion fruit production fields. Fragments from the lesion border were cut and cultivated in the PDA medium, and the isolated strains morphology was observed with optical microscope. The colony color varies from white, pink to purple, and together with the colony characteristic and microscopic structures, the isolates studied are the Fusarium pathogen. And molecular characterization have been done by PCR with the ITS and the EF1 alpha primers. The results found that the 8 isolated pathogen strains were identified as the Fusarium solani, and the other 2 are Fusarium ostiplum. According to Coach's rule, the isolates were inoculated on seedlings by rooting it for the pathogenicity evaluation. We can also find the pathogenicity of the Fusarium solani is higher than the Fusarium ostiplum. It was reported that the cholera disease has been firstly found in China, is in Fujian in 1991, and now it was also spread to almost all the production areas, such as Guangdong, Guangxi, but also in Guangzhou, and Yunnan, and led to the yield with 10 to 60% reduction, and became a huge threat for the passion fruit production. For the integrated control measure, the training course opened on passion fruit cultivation in the countryside for the farmers, for the agricultural technicians of production company or the local government, which will raise their awareness and readiness for the disease. The second is to monitor the disease spread. The rot disease spread in our field is a complicated system through the whole plantation process. Pathogen accumulation in soil or spreads with the plant and soil materials, disease plants, or the irrigation system. During farming, wooding of the corner and root system and waste may spread to the pathogen. It is recommended using the healthy plant material, avoiding planting passion fruit areas with the disease history, and reducing the plant roots and ruling the contaminated plants, then to reduce the inoculum sources. We also found the chemical and biological fungicides show different effects on the strain's growth and spore morphology and their pathogenicity. Anisotropin and anisotropin, zingamycin, and bacillus inoculum showed better effects on the inhibition of pathogen growth. This fungicide were single or mixed to used in field, which resulted in around 30 to 60% control efficiency and would be used to control this disease. For the integrated controlled measure, firstly, we need to understand the mechanism of soil suspension. Fusarium fungus will stay in soil for decades. Organic fertilizer and non-pathogenistic fungus would help to make soil healthier and suppress the disease. Besides, the use of the resistant rootstock have been advocated since it is an effective ecological control measure. The rootstock cultivars was screened out through the disease inoculation and also the compatibility test. Cutting as the perpendicular curtain type were used widely to get more nice and reduce the burden of the trees, which can help to dry the soil and the plant in the rainy season. Eradicating disease plant outside the orchard and disinfected pathogen with the quickening. People will also change the crop production system, such as rotation with the rice, corn, potato, or intercoupling with other crops like ginger, cherry, snake, soybean, or pepper. With these measures, the toxic and harmful pathogen will be diluted and then to reduce the soil suppression. And to screen the disease-resistant cultivars, the germplasm were collected and their seedlings were used for inoculation test and quality evaluation. Resistance breeding program was carried out this year in our team.
Sorry, Dr. Ruibin, you have 1 minute to wrap up. 1 minute to wrap up.
Crossing technology, and now the hybrids were tested in the field test. Some potential lines were created in our project. So in the future plan, firstly, we need to functional analyze the fusarium isolates. And it's vital to better understand the genetic variability, genetic diversity, or for the characterization of these isolates. Secondly, to understand the plant fusarium interaction mechanisms at gene, psychological, and molecular level. And last, to develop and apply appropriate diagnostic surface with the intelligent recognition. So the breeding program in breeding program, resistant genotype was used as the parental lens for the crossing. And earlier screening assay and the resistance evaluation methods should be established. And then the case 9 and multiple gene editing have been used successfully in other crops, but in the passion fruit, it still has a long road. So that's all for my presentation. Thank you, Dr. Wei's lab in our institute and Dr. Lee for his help with the disease research. That's all for my presentation. Thank you for your attention.
Thank you very much, Dr. Ruibin, once again. It is clear from the two presentations we had today from China, tremendous strides have been made in terms of the country's production and trade of passion fruit, despite looming threats such as the presence of disease, which, as shown by Dr. Ruibin, are being continuously researched and monitored, and the development of breeding programs for resistant genotypes and integrated control measures which are in the pipeline. Thank you very much, Dr. Ruibin. We now would like to welcome our final speaker for the day, Dr. Win Van Viet. Is Dr. Win online?